last lecture for Unit 2's examination of biological psychology. And in this lecture, we're going to look at the methods that psychologists, scientists, doctors use to study the biology of humans. So in terms of methodologies, the most ancient of the methodologies is dissection of the brain. The drawback, of course, is that that can only be done on the dead. So we are not seeing a living process as it is happening, but we certainly can see the structures that are there when we do dissection. And even our ancestors noticed this when there were head wounds and lesions and breaks in the skull. They could look in and poke around and see what happened. In the United States, there's a very famous case of a man who had a rod blown through the front of his face and came out the top of his skull. And obviously, the wound provided some data for doctors, albeit they kind of didn't know what they were doing then. But as more and more doctors and neurologically minded folks were examining human brains on the dead, it did teach them something about what the structures were. The only problem is, of course, those people are dead. That does lead us to another part of the methodologies that we can use for learning about the human body. And that would be destroying or burning away part of the brain. This is called ablation. You can also make a lesion in a brain. You can cut out parts. And when you do this, you can see what happens to the person after you make the ablation. Obviously, that's a pretty invasive thing to do, and the effects could be catastrophic on the person. So typically in the past, this would have only been done for people in extremely problematic medical conditions where, you know, their quality of life was already pretty limited. And therefore, this was a very radical way of trying to help them at least survive or have some measure of living that would be bearable. It's not until the 20th century that we get more sophisticated techniques other than cutting holes in people's heads. And one of the first ones is a CT scan. A CT is essentially an X-ray. The way it works is that there are multiple angles of the X-ray, and then those angles are put together to create a 3D image of what's going on in the brain. Particularly good at showing the structure. It's like taking a whole bunch of photographs, putting them all together to sort of recreate what the image actually is. Another one that is useful for not just structure, but function would be a PET scan. In the PET methodology, what happens is radioactive glucose is injected into the body so that the brain will use that radioactive glucose when it is working on and processing information. And you can see where the brain is using that radioactive sugar. So you can follow the pathways of various kinds of activities. You can see how much energy is being used. You can see structurally where that's happening. So these are really great methods compared, of course, to the ancient ones of cracking a head open and seeing what's in there. So we can see structures, we can see a little bit of function, but we can do even better than that. modern ways that we can peer into the brain are MRI and fMRI. Both are extremely detailed. You can see the picture of the MRI here, how much detail you can see. You can identify all the structures that we've already labeled in our own little brain diagrams. MRI is great for just a static picture. fMRI detects blood flow. And so you can watch people solve problems in real time. fMRI will capture where blood is moving. So you can not just see the structure, but also the real-time functioning of the brain. Now, I'm going to leave you with one final thought about all of these. They're great. They're wonderful. But a picture of the brain itself, no matter how detailed, will only tell you what the structures are. You might be able to follow the blood flow. You might even see where energy is consumed. But none of that really tells you exactly how things work. We need not just to be able to see the pictures as they are happening in the brain, but understand the electrochemical processes that are happening, the number of structures that communicate and talk to each other. In many ways, we like to think that all these picture methods will tell us a lot, and yet we still don't know everything just from these methods alone. So being able to see into the brain doesn't actually tell you everything that is going on at the cellular level, at the neuron level, but still a great window into how our brains work and very useful to us in modern medicine, and certainly beats cracking the skull open and seeing what's happening in there. Well, that's been it. That'll be it for our look at biological psychology. I'll see you next time in our next unit on sensation and perception.